All right, so next we're gonna do our experiment where we're using litmus cream, which is just a heavy whipping cream where we mix some litmus powder in. And so this is a pH indicator. So it's at the start here, it's blue and it turns pink if you add acid. And so I'm gonna illustrate that for you by adding some hydrochloric acid into this test tube, and we'll see how it should turn pink. So, and you can see it's indicating that we added an acid. All right, so we're going to do the next part of this experiment. So what I have here are 4 test tubes clearly labeled. It's so important that you keep track of which test tube is getting what treatment. So next, I'm going to add 3 milliliters of litmus cream into each test tube. And so I'm going to try to be efficient here and just do it with one pipette. Stick it all the way down to try to avoid getting anything on the side of the test tube. And then slowly add 3 milliliters. And then move over to the next and add another 3. And the next. And then I'm going to have to refill. So it helps to not go all the way to the bottom of this cream because there could be some particles that could clog the nozzles, but let's see. Oops. All right. I'm going to do the last one. And there we go, 3. So next, I'm just going to follow my table and add the various treatments. So for the first one, we have a pancreatin solution here, and I'm going to add 3 mLs of that to test tube 1. Okay, and I'm also going to have 3 mLs to test tube 3. So, and then the next. Test tube 2, I'm going to add a pinch of bile salts and some water, 3 ml. And then test tube 4, just 3 ml water. So the trick here is to not get the test tube wet on the walls here. So when you add the powder, it all just gets stuck on the wall. So I was trying to be careful with that. And so next, I'm just going to add my pinch of bile salt to test tube 2. And we're going to try to get it all mixed in too. So next I'm going to add water to test tube 2 and 4. And a pinch of bile salt into test tube 3. So next I'm going to try to get the bile salt dissolved into these test tubes. might have to use some paraffin. Actually, I'm going to use a stirring rod. Get it off the walls. Okay. And I am not cross-contaminating my samples. I'm using the clean side of the glass rod in this one. All right, that's pretty good. All right, so next I'm going to walk these over to the water bath, and it's going to sit there for an hour at 37 degrees C. A little thought question: why 37? What does that have to do with our bodies and physiology? All right, so the temperature is 37 degrees, and then I'm going to sit here for an hour. Okay, so here I am taking out my test tube. They've been in for an hour, and we'll just bring them over to our lab bench, and we're going to take a look at what's happened. All right, so your job now is to think about our pH indicator, what has happened to each tube in terms of pH and why that happened, and also look at the color and compare the different tubes. So I'll take out test tube 1. Here is test tube 1. We'll stick test tube 2 next to it. Let's see if I can hold them here. Test tube 3 and test tube 4. Alright, so it does look like test tube 3 is a little bit more pink than test tube 1. And I think if you just look at each of the tube and write down the amount of digestion that has happened and think about what did we put in the tube and how does your result make sense in terms of the various treatments. All right.